Hello everyone, welcome to the latest eLearningLK course offering that we are conducting for this year. And this time we are going to talk about a brand new subject. So this is a very rare seminar that we are intending to do it in both Sinhala and English. And this is a subject that has not been talked that much or as to the extent that it deserves. So this subject is of absolute science, and science is a combination of science and science fiction for me. So I'm so excited to have this opportunity to introduce you to the subject, and most of the people here are already connected may have had some experiences in this area. But this time it's going to be a huge hit, and we are going to introduce Sri Lanka to this subject because it has the opportunity and the potential to propel us into the today's scientific edge, I would say, or the cutting edge of science. So Sri Lanka has been losing such opportunities for many decades to be at the forefront of science, but here we are going to change that, and we are going to introduce the latest on a specific area, a specific scientific area, which is called synthetic biology. So synthetic biology ගැන කතා කරන්න, so we have the best person in Sri Lanka for now, Dr. Lalinka Herath, and we hope to create new synthetic biology scientists in the future through offerings like this. මේ වගේ courses වලින් අපි බලාපොරොත්තු වෙනවා අනාගතයේදී ගොඩක් synthetic biologists ලා ලංකාවේ නිර්මාණය කරන්න. ඒකට පුරෝගාමී වෙනවා Dr. Lalinka Herath. So let's welcome him. මම Sinhala and English mix එකේ යනවා. So you can ask any questions or share ideas at the end of the session from any language, and we will act accordingly. Dr. Lalinka, hi born.
Yeah, I was Sanjay.
අපි මුල් ටික සිංහලෙන් යන්. So මේ බලන කට්ටියට අපි පැහැදිලිව අපි කරලා ඊට පස්සේ අවශ්‍ය තැන්වලදී ඉංග්‍රීසි වලට move වෙමු. එක දෙයක් කියන්න ඕන, අපි මේ පාඨමාලාව අද දවසේ free session එක, but there will be a paid session series after this. Those will be conducted mainly in English, but of course you can inquire with Sinhala language, no issues there. හරි. එහෙනම් මම පොලින්ම අහන්නම් synthetic biology කියන්නේ මොකක්ද කියලා පොඩි අවබෝධයක් දෙන්න, ඊට පස්සේ අපි මාතෘකාවට යමු.
First of all, I would like to thank Sanjay for the lovely introduction, Sanjay. Yeah, ඇත්තටම Sanjay mention කරා වගේ මේ area එක Sri Lankans ලාට අපේ Sri Lankan students ලාට ටිකක් අලුත් subject area එකක්. ඒ කියන්නේ මේක තමයි emerging aspect එක in biology. Biology වල in depth study කරද්දී අපිට එනවා molecular biology කියලා subject area එකක් කියන්නේ we go
තව චුට්ටක් සද්දේ ටිකක් වැඩි කරලා කතා කරන්න පුළුවන්ද?
Sure. Okay, Sanjay.
මයික් එකට ලඟ වෙන්න තව ටික.
Okay, right. අපි molecular biology කියන්නේ අණුක ජීව විද්‍යාව. ජීවය අණුක මට්ටමට ගිහිල්ලා හදාරද්දී අපි කොහොමද දකින අණු වලත් එක්ක ජීවය වැඩ කරන හැටි තේරුම් ගන්න. ජීවියෙ කොහොමද වැඩ කරන්නේ කියන එක molecular level එකට ගිහිල්ලා අපි ඒ mechanisms තේරුම් ගන්න study area එක තමයි molecular biology අණුක ජීව විද්‍යාව කියලා අපි ස්පෙસિෆයි කරලා ඉගෙන ගන්න. ඊට පස්සේ දැන් මේක applications ටික consider කරද්දී මේක applied molecular biology aspect එකක් වෙනවා. ඒ area එකට අපි specifically කියනවා recombinant DNA technology. Applied recombinant DNA technology.
recombinant DNA නේ.
recombinant DNA technology සංජය කියන්නේ අණුක ජීව විද්‍යාවේ අපි ඉගෙන ගන්න DNA පිළිබඳ විස්තර ටික. The information එක අපි කොහොමද manipulate කරලා ඒකෙන් අපි මොකක් හරි application එකක් කරගන්න ඕනේ. ඒ කියන්නේ biology design කරන්න කොහොමද genes වෙනස් කරලා information change කරලා අලුත් application එකක් කරගන්න. ඕක ඉස්සෙල්ලාම touch උනේ මේ applied recombinant DNA technology කියන study component එකේදී molecular biology. ඒක ඊට පස්සේ evolve වුණා genetic engineering කියන තැනට. ඒ කියන්නේ genes අපි කොහොමද engineer කරන්නේ? අලුතෙන් DNA engineer කරලා අලුත් organism එකක් හදන්නේ කොහොමද? ඒ කියන්නේ මොකක් හරි ජීවියෙක්ගේ නැති gene එකක් introduce කරලා එයාගේ ඇඟ ඇතුලේ අලුත් ලක්ෂණයක්, අලුත් characteristic එකක් අපි කොහොමද හදාගන්නේ? ඕක තමයි traditional classical genetic engineering වලදී අපි කරේ. දැන් synthetic biology කියන study area එකට එද්දී මේක තමයි latest aspect එක. ඒ කියන්නේ synthetic කියන්නේ එක අපි define කරගන්නවා නම් සිංහලෙන් ඒක කෘත්‍රිම ජීව විද්‍යාව. ජීවය synthetic biology. biology කොහොමද අපි synthetic විදියට artificial විදියට අපි design කරගන්නේ. මේක synthetic biology කියන platform එකට එද්දී genetic engineering වල latest techniques ඒවා ඔක්කොම combine වෙනවා computational aspects. ඒ කියන්නේ IT related topics එක. Because we have a designing part in this study area, we have to apply the computational tools in designing genes. එතකොට මෙතන වෙන්නේ අපි use කරන එක එක computational tools biology වලදී use කරන අපි tool set එකක් තියෙනවා. ඒක use කරලා අපි DNA එහෙම design කරගන්න. design කරාට පස්සේ අපි ඒගොල්ලෝව අලුත් ජීවියෙක් new organism එකක් synthesize කරනවා that can perform entirely new functions. ඒක humanity එකට benefit එකක් තියෙන function එකක් තමයි අපි perform කරන්නේ. එතකොට synthetic biology කියන topic එක ඇතුළට ආවම not only biology අපිට biology basics fundamentals ඉගෙන ගන්න ඕනේ. ඒ අරෙන්න computational or coding languages and also online computational tools use කරලා අපි කොහොමද DNA manipulate කරගන්නේ DNA design කරගන්නේ ඊට පස්සේ ඒක chemically synthesize කරගන්නේ කොහොමද අලුත් organism එකක් ඇතුළත ඒක introduce කරන්නේ කොහොමද introduce කරාට පස්සේ ආ perform කරන විදිහ අපිට validate කරන්න පුළුවන්. ඒකටත් අපිට computational simulation කියනවා. ඒක කොහොම study කළා AI related latest tool තියෙනවා දැන්. biology එක study කරන්න අලුතෙන් design කරන biological organism එක biology එක study කරන්න කොහොමද ඒක අපිට assume predict කරන්න පුළුවන් මෙහෙම gene වෙනස් කරොත් ඒක මේ විදිහට function වෙයි. ඒවා simulate කරන්න tools තියෙනවා. එතකොට මේ tools ඔක්කොම study කරන subject area එක තමයි synthetic biology. කියලා දැන් latest emerging topic එකක් study area එකක් විදිහට propagate වෙලා ඇවිත් තියෙනවා. එතකොට අපි මෙතන gather වෙලා ඉන්නේ synthetic biology කියන area එක ගැන special focus එකක් තියාගෙන කතා කරන්නේ.
Okay. දැන් මගේ channel එක බලන කට්ටියට නම් ඔබ අමුත් එක් නෙමෙයි. නමුත් අලුතෙන්ම මේ audience එකට සම්බන්ධ වෙලා ඇති අයගොල්ලන්ට පොඩ්ඩක් හැඳින්වීමක් කරමු වාගේ background එක.
මගේ background එක කියන්නේ මගේ educational background එක ගත්තොත් සංජය මගේ ආචාර්ය උපාධිය PhD එක කියන්නේ genetic engineering වලින්. ඒ කියන්නේ ජාන ඉංජිනේරු විද්‍යාව. ඊට කලින් මම මගේ masters උපාධිය හැදෑරුවා ඒක කියන්නේ molecular genetics කියන area එකෙන් තමයි. මගේ ප්‍රථම උපාධිය මම නිම කරේ කොළඹ විශ්වවිද්‍යාලයේ අණුක ජීව විද්‍යාව හා ජෛව රසායනික විද්‍යාව කියන ශේෂ්ත්‍රය. ඒක ගොඩක් fundamentals ඉගෙන ගන්න degree එකක් molecular biology related. ඒක මම narrow down කරගෙන ගියේ genetic engineering side එකට PhD එකේදී එහෙම මේ thesis එක specify වෙද්දී. ඒ අරෙන්න මගේ experiences ගත්තොත් මම ගොඩක් synthetic biology related research groups එක වැඩ කරා ඔස්ට්‍රේලියා වල ඩීකින් සරසවියේ synthetic biology research group එකේ වැඩ කරා මගේ ආචාර්ය උපාධිය කටයුතු අතරතුර ඒ අරෙන්න University of Granada Spain CRISPR gene editing group එකත් එක්ක වැඩ කරා ඒ කියන්නේ gene edit කරන්න use කරන tool එකක් study කරා ඒක CRISPR-Cas gene editing tool එක කියලා ඒක ස්පේන් වල University of Granada වල තමයි ඒ පිළිබඳ කටයුතු හැදෑරුවේ. ඊට කලින් මම මගේ මාස්ටර්ස් එකේදී මම වැඩ කරා සවුත් කොරියා වල ජෙජු නැෂනල් යුනිවර්සිටි එකේදී. ඒ අරින්න දැනට මම රිසර්ච් කෙනෙක් විදිහට ඇටච් වෙලා කොළඹ යුනිවර්සිටි එකේ Center for Advanced Material and Devices can be synthetic biology related research conduct කරනවා මගේ ස්ටුඩන්ට්ස් ලා එතන බේස් වෙලා synthetic biology research වල ඉන්වෝල්ව් වෙලා ඉන්නවා මේ වෙනකොට ඊට අමතරව මම CSO විදිහට වැඩ කරනවා අපේ ශ්‍රී ලංකාවල අපේ පරණ ස්ටාර්ට්අප් එකක් Torrington Genomics and Bioinformatics කියලා කොළඹ යුනිවර්සිටි එකේ ස්පින් ඔෆ් එකක් ඒක අපි වැඩ කරගෙන යනවා ඒ පැත්ත ජීනොමික්ස් කියන ස්ටඩි ඒරියා එක මේන්ලි එකේ ටච් වෙන්නේ ඔවා තමයි වැඩේ අරින්ද
සෑහෙන ඇක්ටිව් ලයිෆ් එකක් තමයි මේ දවස්වල ගෙවන්නේ ඔබ එහෙනම් මම හිතන්නේ ඒ පරීක්ෂණ කටයුතු වලටත් අපේ සිසුසිවියන්ට මේ පාඨමාලාවට සම්බන්ධ වෙන කට්ටියට යම්කිසි ඕපනින් එකක් තියෙනවා මම හිතන්නේ ඔබ ඉස්සරහට ඒක ගැන කතා කරයි. So I leave the stage to you to start the presentation. Everybody stay put and we are going to bring you some good news at the end of the webinar as well. The webinar එක අවසානයේදී අපි ඔයගොල්ලන්ට හොඳ offers ටිකකුත් අරන් එනවා. මේ වගේම පාඨමාලාව ගැන අප්ඩේට් ටිකකුත් දෙනවා. ඒ වගේම අද subject එක ගැන ඉගෙන ගන්න Dr. Lalinkaගේ first session එකට එන මෙන්න අවසර.
Okay. Thank you very much, Sanjay, for your. Yeah, thank you. Let me first share my screen. Okay. Very good evening, everybody who's joining here this evening to hear some interesting topic on in biology, I would say. So um I'll just keep it simple because I want you all to get a simple idea about synthetic biology because this is the first session that we are having together. Right. I'll start it like this. Let's say I was just wondering, the students who are joining here today, uh, are you someone who's inspired by science, see how the extinct organisms like dinosaurs was brought back to life. That's how actually the interest in me triggered in science, right? And also, have you seen the movie Planet of the Apes? The apes evolved into certain stage that they can even compete with humanity. And also, did you watch X-Men that human actually gain superpowers? And one popular movie, Spider-Man, where a human gain the attributes of spiders. And one of my favorites, Assassin's Creed, where the genomic information in a human uh, is triggered, and then his ancestral memory is activated, then he becomes an assassin. So all these things link to a certain area that we are interested in, which is the genetics and also DNA. So we wonder whether these kind of things, everybody, this is the ideal platform for you all. This discussion is the ideal place for you all to have a chat on and also explore whether these things can actually be done in real world, right? The topic we are going to discuss today is synthetic biology. The biology is created. Whether we can recreate the biology is what we call synthetic biology. Right. There's a saying that synthetic biologists can play God, which means synthetic biologists are supposed to be changing information in biological systems. Therefore, some refer to them as God. So let's explore and see whether this statement is true or not throughout of our session today. Right. Right. I'll keep it simple. Now, to understand this topic, we can just think about animals, the day-to-day animals that help us in day-to-day activities. Think about cow who provide us milk, and also you can think of horse. So they support us in traditionally they supported us in transportation, and there's lot other animals you can think of who help us in various aspects in our day-to-day life. And also, you can't forget about plants in nature, that plants can also help us in lot of ways in day-to-day life. So they provide us food mainly, and also they provide medicine, and you can use them in many other as utility in our day-to-day activities. So these are part of nature, the animals and plants. Uh, Naturally, they actually help us in several aspects in symbiotic manner to sustain our life as humanity. Now, as synthetic biology, into the topic of synthetic biology. So they anyway help us, but as synthetic biologists, we can think of contributing into the enhancements of these attributes of animals and plants that can help us in certain day-to-day activities. So by the definition, the scientific definition, synthetic biology is what? So you define this area as the designing new biological organism. Basically, you can design, redesign, you can create biological organisms. For what? For environmental and also for health related application. So this is what we call synthetic biology. Basically, the scientists in synthetic biology can redesign re-engineer biological system in order to provide us with beneficial functions. Right. So area is defined as known as synthetic biology. So how can you actually real world applications? How can you do this to redesign? In designing, you have to initially program these animals and also plant cells. To do this, we have a set of tools, guys. These are known as gene editing tools, and the area is known as genetic engineering because we are going to change the information in biological systems or engineer information in biological systems. Right. So these the certain tools that we use as synthetic biologists and theories, techniques that we apply in this particular area is what we are going to explore here together in this session together because we want to understand how these exciting and also interesting applications are actually being done in scientific world, right? Uh, right. I believe most of you who join here today has certain sound knowledge of biology, in biology, basic biology. Therefore, you know the human, plants, and also the other animals in nature, they are built up of our unit of life is cell, right? We are built out of cells. Inside of cells, we have an interesting component or organelle. We call them organelle. Organelle called nucleus. So in nucleus, that's where the material, chemical material DNA is located. In order to change information, we need to identify where the informations are being stored. So that's inside of nucleus and in DNA. All right. Now think about DNA. To understand how DNA works, you can refer to this analogy. You can think about DNA as a book written in a 4 letters language. What are the letters involved? A, T, G, and C. Based on the combination of these letters occur in DNA, a certain information can be stored in a DNA molecule, which means, as an example, let's say information about animals or human means our hair color, our eye color, the way I talk, the height of a human, right? All these things are humans' attributes. So these attributes are different from one person to another person. Those information should be written in a certain language. That's what we call as the DNA's language, with 4 letters language. Let's say my hair color is brown and your hair color is black. So the information to explain or mention, communicate this information is written in DNA's language, a specific segment in DNA where this different information are being stored is known as a gene, right? So this you learn in your high school classes as well. So I just wanted to simplify this concept and share with you to understand how information are being stored in DNA in the form of genes, right? Right. How this information storing itself is not enough in order to determine the certain attributes in organisms. This information needs to be expressed whenever required. To understand that, we have to refer to one of the core concepts in molecular biology, which is known as the central dogma of life, everybody. Which means we have genes inside of nucleus, and genes need to express this information whenever required. So that happens via a two-stage process known as the central dogma of life. The first step of this process is known as transcription. There's a time we talk whether transcription need to be the first step, and then second step is translation. How the mRNA molecules are being made out of genes information in DNA, and then protein are being expressed, which is protein. Proteins are the certain component that perform function inside of our body. So expressing protein or producing proteins is the fundamental aspect of doing certain biochemical reactions inside of animals, plants, body. So if we could change now in this case, if information is stored in gene, if you could change the information in a gene, you can ultimately affect the nature of the protein that is being coded by that particular gene. Idea is that you can just use a certain gene editing tool in order to change the information in a gene and come up with a modified gene, right? Once you have a modified gene, this the changed gene, or you could call it modified gene, can express entirely new modified proteins that perform entirely new functions inside of biological system. That way you can actually engineer the information inside of biological system. So this is the core concept in genetic engineering, which leads to synthetic biology eventually to come up with bio entirely designed or but newly designed biological system. Right. Now, when it comes to the theories of synthetic biology, what are the principles that are applied in synthetic biology? There are four main aspects in synthetic biology. Because in synthetic biology, idea is to come up with new biological organisms that can act entirely in different manner from the natural organisms. So these biological systems you have to initially design. So we have a design stage initially in this particular study area. And then once you design this using certain tools in synthetic biology, you can then build that particular organism, right? Once you build this organism, since this is a new organism, you have to test them for their activity or their function. Let's say you want to change certain information inside of biological organism. Think about plant that you want to make it pest resistant. Naturally, this particular plant is not pest resistant. What you could do is you can introduce a new gene into this plant that expresses protein which is toxic to certain pests, right? So whether it actually expresses this particular protein can be studied once you engineer it and build it with certain stimulation tools. You have to test it for this novel function in that biological system. And then you learn it. In the final stage of synthetic biology projects, you have to learn how these newly designed biological systems behave. So these are the fundamental aspects of or main components of synthetic biology related projects or synthetic biology research. Right. Right. I will briefly introduce some of the tools. I know these sound technical, but we can get an idea how you can actually do these things initially. To design, we have a set of computational tools that are very much user-friendly for biologists. As I know whenever I mention the word computational tools, the biological science students, they are like a bit scared of computational tools. But don't be scared, guys, because these tools are designed for biologists. So you can use them in the tools like Benchling, Tinkercell, SBOL Designer. So using these particular tools in synthetic biology, we can design new genetic systems inside of biological system in order to program the living cells. Once you design it, you have to build it by using certain tools like J5DNA assembly design, genome compiler, NetBuild assembly tools. So there's all list of tools that you can use in building these biological systems that we have optimizing synthetic biology. Right. And in order to test, we have tools like MATLAB, Symbiology, and also finally to learn how these newly developed biological systems behave, we have the popular tools like Copasi and Tellurium, SLBL compatible toolkit. So whole lot of things that we can explore under the synthetic biology topic, how we can actually create new organisms and then test them and validate their performance. Right. That's a small insight about the technical aspect of synthetic biology. And also we can explore the applicational range of synthetic biology as a study area, mainly as biomedical science students and also biotechnology students. We want to understand how this knowledge can be applied into certain areas. Now think about two main aspects when it comes to biomedical sciences and also biotechnology. Our idea as scientists in synthetic biology is to find solution for health-related matters. Health-related in the sense, there are two aspects. We need to develop diagnostic tools, disease diagnostic tools. At the same time, we need to develop new therapies for certain diseases. So these therapies that we design under synthetic biology techniques, utilizing synthetic biology techniques are known as engineered therapies, which mean as an example, let's say there's certain cancer condition that you are interested. You need to kill these cancer cells, right? Naturally, our immune system has a certain cells uh, that can target these cancer cells and kill them. Hence, in this attributed certain immune cells that can kill cancer cells. What you could do is there's a certain cell type called the CAR T cell in our immune system. You can use synthetic biology tools in reprogramming these CAR T cells in order to target the cancer cells. Let's say it's breast cancer that's been growing, you want to target breast cancer cell, right? In that case, you can engineer the DNA inside of CAR T cells into patient's body and target these breast cancer cells in order to kill those cells in breast cancer patient. Also, we can engineer like microbial systems that can early detect the breast cancer biomarkers, which means you redesign a microorganism that can capture the biomarkers. You learn biomarkers in your biological topics in first one or two sessions, years in biomedical science students mainly. So biomarkers are certain indicators of certain disease conditions. You can design microorganisms like systems in order to capture this biomarker and emit a certain light signal that can signal for the presence of this particular disease type in a patient's body. So that's also one of the interesting applications related to synthetic biology when it comes under health. And you can produce engineered drugs. We will learn those things as well under synthetic biology topics. So think about environmental application. So this is for biotechnology students, guys. Bioremediation is one of the major aspects in environmental sciences, which means how you utilize biological systems in order to remove pollutants in certain environmental polluted sites. Right. Think about um, a certain water body, like in ocean, there's an petroleum oil spill due to a leakage from a ship. Right? You want to effectively remove this petroleum spillage. What you could do is you can develop new microorganisms by changing their DNA and engineering a new synthetic genome system inside of this particular microorganism in order to eat this petroleum compound and degrade it down to environmental friendly compounds. So these are the green aspects. So I don't want to take more time on elaborating these aspects since we can discuss them in a certain study component when it comes to actual phase of learning these tools. Right. Not only those applications, you can think of some interesting application like pet cloning, whether you want to clone your pet. Everybody loves their pet and they want to clone. We have the set of tools that are improved in the area of synthetic biology that we can actually clone our pets. If you read about these areas, you will come across there are companies in the world that provide pet cloning services. They basically use these tool sets in synthetic biology. And the glow in the dark plants you can make. And usually the plants, the higher plants, they don't glow at night, but you can introduce proteins from different organisms like firefly protein, and also if it's bioluminescent, you can extract from certain organisms and then make it express inside of plants in order to grow them at night. And you have seen, if you're a pet lover, fish lover, you know that in aquariums you can purchase this commercially produced genetically engineered fluorescent fish that glow once you expose them to UV light. So these are some of the interesting applications. And there are emerging applications like DNA-based data storage. Now the scientists the experts in information, IT field are trying to store certain information in DNA because DNA is a language where information can be stored. And then you can, instead of storing information in CD drives, USBs, pen drives, hard disks, you can store information in DNA. And programmable cancer vaccines is another topic. There's lot other thousands can utilizing these tools, you can come up with new strategies to find answers for burning issues. Right. Right. Now, since we talk about this interesting application, I know you are wondering whether these can be actually done in a scientific uh, setup. Just to let you know, even though these are nerdy stuff, a bit of uh, technical aspects, share a bit of technical aspect, I would like to share the actual workflow of doing designing a synthetic organism using synthetic biology techniques. Right? So as we mentioned before, there's something called genes inside of biological system where the certain informations are being stored. So we want to initially identify what are the genes responsible for different characteristics that you want to change. So the initial phase is to identify these genes in certain databases. And then you have to validate these genes using certain bioinformatics tools that have been optimized in under synthetic biology topics. And there's another principal technique in synthetic biology known as preparation of logical framework, which means how you can logically contrive the expression of genes inside of a biological system and designing a model for simulation. Before you test this biological organism, you have to design something called a biomodel. And this model is what you have to test for certain interesting applications. And then the identification of host receptor compatibility, preparation of genetic logical structure, finally to simulate to a test organism. And here's how you actually develop a new organism in synthetic biology in step by manner. One of my previous undergraduate students work on developing a genetically engineered or synthetically designed microorganism to detect microplastic in contaminated water. So these are the tools that he used in designing this organism. Initially to identify these genes that are responsible in degrading microplastics. It's a big topic now when it comes to environmental pollution. So we want to find a solution in come up with a new organism that can eat microplastic and then remove from contaminated water body. So he initially mined these genes responsible for microplastic degradation in GenBank, NCBI GenBank using certain basic tools in bioinformatics like BLAST tool. And these tools you learn in first your undergraduate topics. I know BLAST tools and all. And then, so these are the specific tools that you haven't touched before. How you can actually, once you know the genes, how you can actually design the presence of these genes inside of a biological system. So in here, we apply one of the interesting theories in synthetic biology, which is known as genetic logic gates. And how you can, similar to what you learn in physics that known as electronic logic gates, once you provide some input into biological system, it should give us an output. So we can comprehensively learn these things in our synthetic biology courses if you are interested in joining in future, guys. So these are the concepts we are actually going to explore and study, learn in these courses, guys. Once we provide inputs, let's say there's two input, A and B, which means you can think of two pollutants. You want to detect the presence of heavy metals, cadmium and lead, using a genetically engineered bacterial organism. You provide A as cadmium, B as zinc, and then Z is the outcome. You need a fluorescence signal to sense for the presence of element. So these are called the logical construction of genes. So to perform this actually inside of biological organism, you have to introduce these different genes into biological system and arranged in certain manner that they are being activated one after another in the presence of these particular inputs. Right. So this is the logical framework designing part of synthetic biology. And then you have to come up with something called biomodel. That's what you are going to simulate computationally and see whether your biological system, newly developed biological system, functions as per the way that you expected in the designing part. So in order to do that, initially you have to design something called the biomodel of that particular biological system. That is being done with certain tools known as Tinkercell and also SBM sequence. These are the tools that you need in designing biomodels in synthetic biology. So we can learn them in synthetic biology topics, guys. So, and also to find out, this is one of the computational biology topic. Suitable host receptor identification need to be done. That I don't want to go into technical information in this particular stage. That's later we can explore and learn these tools, how we can utilize these techniques in designing maybe certain therapies for cancer. Right. Yeah, these are the actual data my student produced in the laboratory in order to understand how certain inputs can interact with receptors of the biological cells. And then here is the biomodel that he designed uh, in order to simulate or redesign, engineer a new microorganism that can use the microplastic. And then here's the genetic logic gate framework. So in this aspect, I'll try to simplify as much as possible in this quickly. Now look at this diagram. Now there are three inputs, PET, PE, and PS. These are enzymes, guys. Once the presence of a particular microplastic type like PET, polyethylene, and one of the other type, the certain signal, blue fluorescent protein signal is activated inside of this microorganism, what he has designed. And then once it is activated, this newly designed microorganism is capable of expressing enzymes that can eat three different types of microplastics. So that's how he constructed this genetic logic in order to dictate different uh, types of microplastic. Now look at this. This is the test simulation part. To use Popasi-like tools, simulation tools, in order to understand how this particular microorganism actually behaves if you chemically synthesize it in the laboratory. So you can see how the particular genes are being activated and then how the certain genes are being activated. Each of these plots the level of expression level of different microorganisms. Right. So that's some technical knowledge I briefly shared with you. We can later learn a comprehensive manner in technical courses. Right. So I want to share the scientific research that we have conducted in our research group related to this study area. So this particular study published in BMC Biotechnology is one of the international journals, and here what we have done is we have developed a new genetically engineered microorganism known as gem biosensors to detect heavy metal in contaminated water samples. Right. So these are the biomodels we have designed in this particular research, and we have published that in certain journals. And also, not only that, we did not stop at that point. We also extended our research in integrating that with electronic aspects. Once we have developed this biological system that can emit fluorescence signal in the presence of cadmium, lead, and zinc like heavy metal. So we want to detect that signal utilizing a certain heavy metal detection system. So you have to actually collaborate because this is a multidisciplinary study area. In synthetic biology, you have to collaborate with the certain expert in other areas as well. That's why we collaboratively work with the electronic engineering group and develop a portable electronic detection system that can capture fluorescent signals emitted by this genetically engineered microbial system. And we recently received the patent for this particular heavy metal related signal detection system as well. So I can present this as one of the first synthetic biology related commercial projects that has been done in Sri Lanka in recent past. Right. So this is another study conducted by our research group that we have small fish, fluoresce, a red color signal in the presence of heavy metal in a water sample that this fish is been swimming in. Now look at this. Once you see, can you see the red color signal in fish? Because this fish has been genetically engineered in a manner that it can emit a signal in the presence of heavy metal once the fish is treated with heavy metal contaminated water samples. So even the workflow of this particular in design our topics under synthetic biology courses. Right. Now we know that changes can be done in organisms using synthetic biology tools. But are we actually supposed to change information haphazardly in biological systems, guys? That's something you have to concern when it comes to synthetic biology research. Even though we have the set of tools that we can change information in biological system, this has to be done very ethical manner, which means we are not supposed to edit certain genes that affect in adverse manner in biological systems, in the nature, on the nature as well as in organisms. We should not change information in immune systems that are not very well studied. So these are regulated by research ethics. Therefore, in synthetic biology, we have a major component. If you are a student in synthetic biology, we have a major dedicated area to learn how you actually utilize these synthetic biology tools in responsible manner in order to design new biological system the benefit for the benefit of humanity. So that also we can explore in synthetic biology topics. Right. Finally, I would like to share this. Yeah, the ethical aspect is very important. If you watch the TV series like Biohackers, I don't know if you have seen this. This is a very interesting Netflix TV series that showcases what happens when you unethically edit genes in humans and the consequences of gene editing in human. It's I recommend all of you to watch this if you are. I know the students joining are science enthusiasts, so you all should watch these kind of TV series and also learn how these plots are being inspired by the bioethical aspects and also the synthetic biology techniques. So this is one such TV series I can recommend for you all to see. Okay. Finally, yeah, as you can understand that synthetic biologists can change informations in biological organisms, I would like to refer a quote by famous synthetic biology who's Prof. Craig attached to MIT that he always mentioned that we are not playing God, we are playing the software engineering of life because, as I mentioned, the nucleus and the DNA is the IT unit of biological system. We are basically changing information in the DNA in order to construct new information in biological system. God, yeah, basically pretty much the same you would say. So it's plain the software engineering of life. Synthetic biology is basically. For the first time, a living organism's parent is a computer. Because Craig is the person who first developed a synthetic genome. So this is the quote he came up with in order to refer, popularize this study area among scientific world, guys. Right. Okay, so it comes to the end of the certain insight on synthetic biology application and also the synthetic biology related techniques that you can utilize in designing biological organisms. And the final idea was to yeah, share. We were in the discussion of developing a new synthetic biology course in Sri Lanka, which has not been done before. So that we are going to provide through e-learning platform for our students to explore the latest techniques and the emerging aspect of genetic engineering, recombinant DNA technology, and molecular biology, and how those are being integrated in designing synthetic organisms. So it's going to be a 1 month uh, short course that in the initial phase. Sanjay, are you around?
Yes.
Yeah, Sanjay, you want me to continue with the short course content, or are we taking a break and then move on to that part? So what's the plan? Yeah.
Okay, I'll explain then how to connect with our course going forward.
Yes.
Then you can take it from
Right.
there.
Okay, sure. Okay. Over to
Okay.
you, Sanjay. Right.
Okay. Okay. So you might be wondering now, okay, I'm interested in subject and I understand what Dr. Lalinka is going to teach us, and let's see how to connect with the course itself. So you just have to keep in mind that we are going to start this course. It's a 4-week course. I mean, it's 16 plus hours. It's going to be over within 1 to 1.5 months. So it's going to be a quick finish. Maybe within July, we will finish the course. We are going to start it on the 2nd of July. And if everything goes well, we will finish the course within July on July 27th. So it's every Monday and Wednesday, 7 p.m. to 9 p.m. So each week you will have 2 days, 2 classes. Monday and Wednesday, 7 p.m. to 9 p.m. Medium will be mainly in English. And if you want, you can ask questions in Sinhala. So you can get unlimited access to class recordings for 1 month after the course ends. So even if you miss a live session, doesn't really matter. There are a lot of, we provide the facilities through our LMS to access our recordings. And then you can stay connected with the teacher, with the lecturer anytime through our private WhatsApp group. There will be a separate WhatsApp group for the people who are joining the class. And all student live sessions who are joining will get a participation certificate as well. And there will be an exam at the end. So once you complete that practical exam, you will earn a digital credential. So digital credential is a globally recognized credential where you can display it or give a link to it via your LinkedIn or any other CV or resume that you're having. You can integrate it into that, and the digital badge is third party verified. The same digital badge, Meta, University of Chicago, all those people are giving that. So we are having the same technology, the digital badge offered by Badger and Canvas Badgers. So what you will learn, so Dr. Lalinka has already explained and he will explain further after this. But I will, in a very brief summary, I will say week 1, foundation of synthetic biology, week 2, environmental and agricultural applications, week 3, healthcare and therapeutics, week 4, computational tools and project design. So you might be wondering what is the course fee. So we are eager to keep it. I mean, we have always strived to keep unique knowledge like this affordable to everyone. So it comes at a price of 25,000 rupees. So you have to pay 25,000 rupees to get into this course, but there will be a discount. There's a huge discount actually. There's a 20% discount, reducing the total fee to 20,000 rupees. So this is valid until 4th of July. So if you get into the course before 4th of July, then you can get a 20% off. So you will get the course at 20,000 rupees, but you have to make the payment at once. So it's only for full payments. So full payments before 4th of July, you will get a 20% off discount, making the total course fee to 20,000 rupees. And then you have flexible payment options. If you cannot pay it upfront, then you can have 2 to 3 installments based on that. So basically, if you have Mint Pay, you can go up to 3 installments, but you have to have the balance. So when you are paying in installments, you have to pay the entire 25,000 fee. So no discount is available if you are paying in installments. And then always join our WhatsApp group. If you haven't already, I'll just put the link after this. So you can join our WhatsApp group. All our updates and everything will be done through that. So to recap, we are starting the course in the 2nd of July, and then you can join the course immediately right now. The offer is open. So if you are paying upfront, you will get a 20% off. Otherwise, you can pay in installments, and it is going to be 25,000 rupees. And you can do payments with the various credit cards, bank apps, checks, and even bank transfers. So everything is available, and we will be here to help you. And if you have any questions, do ask in the WhatsApp group, or you can call us on our hotline, not 756 700 700, not 756 700 700. All those information will be up on the Zoom chat as well as the YouTube chat right now. So before Dr. Lalinka starts his session again, I would like to request you to fill up the feedback form. Now it will come through the Zoom. There will be a poll, live poll. Please mark the poll and tell us what you feel, what's your feedback on this particular session that we have done. Okay, Dr. Lalinka, back to you, and I hope to see most of you on our first session on the 2nd of July. Hello, we can't hear anything.
Yeah. Okay. Right. I'm here. Yeah. Okay, Sanjay, thank you very much for sharing this information. Now, yeah, I would like to quickly glance the outline and how we have planned the course here as well. Yeah, we have this online course specifically designed for the biomedical science and biotechnology students. In the initial phase of the course, what I have actually planned is to provide our student with the knowledge on basic fundamentals in molecular biology and how the applicational component, the applied aspect of molecular biology is molded inside of recombinant DNA technology topics, and then how it is being integrated into evolved to the stage of genetic engineering techniques. Eventually, how these techniques in genetic engineering, recombinant DNA technology can also be reinvented in synthetic biology platforms by combining it with computational techniques and also mathematics tools and also engineering topics. So that's the idea of how what synthetic biology means will be studied in the first phase of our course, and then how it is integrated into how you can use these fundamentals in synthetic biology in certain environmental agricultural applications and also healthcare therapeutic applications. And what I'm going to do is I'm going to together study with my student the actual workflows of designing microbial biosensors for environmental applications, how you would actually design biological systems to bioremediate certain pollutant in the environment, how to bioengineer a crop using synthetic tools, and how you can design genetic logic gates, genetic circuits as diagnostic tool in healthcare. So I'm going to go to elaborate these workflows in the laboratory and also computational tools that you are going to use, how to actually utilize computational tools in this particular application will be studied in the second phase of our course. Finally, I want my students to work on their tailored project, specific project related to synthetic biology. They can come up with their own ideas and then we will incubate them in order to design their gene circuits, genetic circuits related to their idea. And finally, how you would come up with certain gene circuit that you can write a research article on it and publish in an international journal. And also how you would patent, claim the patent for that particular new biological system that you are going to design. And once you patent it, how you can commercialize it. So that aspect will be studied within the final stage of our project, our course, and then we will guide students through this particular process of publishing, patenting, and the top 5 performers. I just wanted to mention that we will select the top 5 performers to their project, and then we will take them on board in order to work long term with us as part of our synthetic biology startup company that I don't want to reveal the name right now. This will be the first synthetic biology related company based in Colombo, Sri Lanka. So you will be part of this interesting project, historic project, guys, if you perform well throughout our course. And these are the things that you gain by joining our groups. You will definitely get hands-on synthetic biology knowledge, how to utilize these computational tool in designing biological system. And as Sanjay also mentioned, you get to a valuable certificate, digital certificate, and e-badge via eLearningLK. And we are this is going to be a lifelong journey to join with us, and we want you all to work together with us in our ongoing research and also innovative projects that are aligned with our startup, coming startup and industrial opportunities, how you can explore industrial opportunities. One thing I want to mention, the students are curious about what sort of job roles they can be in, uh, they can find once they complete these kind of courses. Actually, we are not much knowledgeable about the course, the certain job roles under this particular topic. I must say, now there are emerging job roles like genetic circuit designers. Some companies advertising, foreign companies advertise, and they are eagerly looking for the genetic circuit designers to be part of their R&D uh, section of the company. So you can apply for such job roles as well if you are interested in migrating, and also you can become the synthetic therapeutic designers, one of the major emerging job role, and you would get very well paid, like if you can become a proper synthetic therapeutic designer, like you can patent your gene circuits related to cancer therapies, and then you can commercialize it, earn millions by patenting your certain therapies, guys. Of course, we will recommend our student for postgraduate studies. One of the major main interest of our students after the completion of their first degrees in biomedical science and also biotechnology. As I mentioned before, also we want you all to work with us in our project on developing the Sri Lanka's first, I would say, to the best of my knowledge, that would be the first synthetic biology related startup. And our students are currently actively working on developing the foundation to initiate initial operation of this particular company. I don't want to reveal the name of the company right now. Later we can uh, share that information as well. Right. Finally, but not yeah. As a practice, I want to thank all the institutions where I worked, where I studied, like Colombo University, Deakin University, University of Granada, Jeju National University, where I actually accumulated this cutting edge knowledge, and the giants in our field, and who first edited the higher vertebrate organism in Sri Lanka, the late Professor, Senior Professor Praharis Dasanayak, and Professor Nalinda Silva, who supports us in popularizing this study area. Even if Professor Nalin is a nanotechnologist, he supports us a lot in popularizing this synthetic biology research area in Sri Lanka. And the certain teachers who trained me at Deakin, Prof. Luis Afonso, Professor Federico at University of Granada, Professor Lee at Jeju National University, and our current institutional partners can be Center for Advanced Material and Development at Devices at University of Colombo, and also NIC, the National Institute of Japan. I want to especially mention them because they generously help us in initiating first genetically engineered research project in Sri Lanka back in 2017 with our transgenic zebrafish project. And NRC, National Research Council Sri Lanka, supported our initial projects in developing synthetic microorganisms for heavy metal detection, and my current research group I want to mention as well. Right, that's about it, guys. So we can discuss if you have any further question and your interest, what sort of things we can clarify related to this area. Thank you
You
very
can now
much.
unmute your mics and ask
Thank you,
questions,
Sanjay.
or you can use the chat.
Yeah, I saw this interesting question in chat, which popped in chat, Sanjay. Let me check from one of our, someone asked, it's not there anymore, I don't know where.
Okay, you can put the questions into the chat, or you can unmute the mic and directly ask.
Yeah, that's a computational. Yeah, I'm working in IT industry, someone says, and he's interested in this particular topic. Yeah, definitely. I don't know what's your name, but of course, there are IT students working with us in our projects. As an example, one of my undergraduate students is from the Faculty of Computer Sciences, and he does not have any background in biology. But I wanted him on board in my project because there's a lot of areas to explore in synthetic biology where we have a designing and simulation part of biological systems that we need to develop new softwares as well to simulate newly designed biological system. So this is a very interesting and emerging study area in computational biology as well. So you can later slowly learn what are the applicational aspects in biology, but the tools that are being developed, they are mainly done by the IT people in this industry. So I definitely recommend you to join our course and explore these tools in synthetic biology and see if you could come up with new software that we can simulate newly designed biological system as your project. Yeah. And DNA-based data storage is also one of the interesting aspects that IT students can explore. Right. Any other questions?
Yes, this is totally online, 100% online.
Yeah, of course, Varun. Yeah.
You have to access it through eLearning.lk. Once you register for the course, the link has been given already. So you can register online, directly pay and register, or you can do a bank transfer. Once you've done that, we will give access on the day. So you can access it through the LMS and you will be redirected into the Zoom session. There's a question for you.
Yes, Sanjay, is it in chat? Right. How is bioinformatics related to synthetic biology? Informatics, yeah, bioinformatics mean Fatima, you work with big data sets in databases. So in designing genetic circuits and all, we need to identify the actual matches of DNA sequences and also DNA promoters. So in mining these databases, it is very important to use existing bioinformatics that we know that we refer to as biobricks. Biobricks are what we merge. We link in designing genetic circuits, Fatima. So you have to use certain bioinformatics tools in mining the databases, in retrieving these biobricks to design bioinformatics, design gene circuits in synthetic biology. And also another aspect, you can also develop new databases. What I feel is that there's a lot of opportunity to develop new databases where you can store annotated DNA information that are useful in synthetic biology application. So one area I recommend for bioinformaticians who are specifically uh, focus on informatics aspect, they can start developing new databases where we can store annotated information in DNA. So we can keep discussing these areas as well.
So what is the basic educational qualification to follow the course?
Uh, yeah, you could be from the background, basic educational qualification in the sense, basic knowledge in molecular biology. That also I'm going to provide in the first section of the course anyway. And also, if you are an IT student, you can also join. There's no restriction for the qualification level. You can be an undergraduate in first year, second year, third year, fourth year of biomedical science, biotechnology stream, and I invite IT students to join this course as well.
And what
Yeah.
about people? I mean, do they have to do biology in the A-level stream?
It's like this, Sanjay. The two aspects I'm going to cover. The biologists should focus on how they can actually utilize this computational tooling uh, in biodesigning their biological ideas. And the IT students, there could be students who do not have biological science knowledge from their advanced level, who are following IT related topics. They can focus on supporting biologists, which means developing databases and also developing new bioinformatics tools, softwares that the synthetic biologists can use. So they can together work. They can focus on two different aspects and learn these topics. Uh, did you understand that aspect, Sanjay, what I wanted to mention? We can the biological science students ලා focus කරන්න ඕනේ මේ user friendly tools use කරලා අපි කොහොමද humanity එකට තියෙන ප්‍රශ්නයකට answer එක දෙන්න අලුත් biological system එකක් develop කරන්න. ඒගොල්ලෝගේ biology knowledge එක use කරලා මේ bioinformatics මේ computational tools use කරලා design කරන්න. එතකොට IT students ලා join වෙන අය ඒගොල්ලෝ focus කරන්න ඕනේ biology තියෙන ප්‍රශ්න වලට biology කල් ප්‍රශ්නයකට උත්තරයක් හොයද්දි මේ බයොලොජිස්ට් කෙනෙක් ඒකට රිලේට් වෙන ඩේටාබේස් එකක් හරි අලුත් software එකක් හරි ඒක ඉක්මනට support කරන්න පුළුවන් software එකක් design කරලා ඒගොල්ලෝ කොහොමද contribute කරන්නේ මේ synthetic biology project එක පැත්තට කියලා. මේ දෙකම අපි discuss කරනවා computational tools section එකේදී.
ප්‍රශ්න ගොඩක් ඇවිල්ලා තියෙනවා.
Okay. Oh, Sanjay. Are gene editing techniques like CRISPR practiced in Sri Lanka? Do we have facilities for such advanced techniques? Also, only a few groups are practicing these tools. We do have facilities, I would say, but not in a single place. Everything is we have to go to one place to another place and get things done. But I mean, in the workflow, CRISPR can be applied. We are using CRISPR tools now. There's one or two projects that I am part of. We are designing CRISPR-based sensing system and also CRISPR-based therapy for multi-drug, the bacteria who are resistant to multiple drugs. So we are going to eliminate that aspect by designing a CRISPR-based therapy in a microbial system. So those are the emerging research, so the currently ongoing research. So we are working on CRISPR research, and we initially need to design these CRISPR-based circuits, and also later we use them. We actually synthesize them with collaborating. We have to collaborate with some foreign companies to chemically synthesize our CRISPR tool. And then what we do is we get it delivered in Sri Lanka. And then we can use those tools in our wet laboratories. We have ongoing projects, one or two ongoing projects. If you are interested, you can reach out to our group where we can share some information on how we can actually plan the CRISPR-based research base in Sri Lanka. I'm undergraduate, BMS, MSDM. When are we going to start on our projects? That's the course we are going to start in July 1st month. In the 4th week of our course, you can start your own project. And once you within 2 weeks, you have to finish the conceptualization part and also the simulation of your gene circuit. Once you learn the tools, you can do that in 2 weeks. Once you are done with that simulation of the gene circuit with computational tool, we will select your project and then see whether we can proceed with the further aspects of your project in order to patent your work and also commercialize it. So that's the plan with this course. In the 4th week, we will start your project. After completing this course, what are the career paths available in Sri Lanka? Good question. There are certain places you can work and initiate new projects. I don't. You can later reach out to our group. Then I'll mention the institutes, names of these institutes, where you can be part of in the R&D company, R&D division, and then start proposing synthetic biology related therapeutics and also genetic circuit designing components. Now, even in research, you have a lot of opportunities in Sri Lanka. We are gaining a lot of funding into these kind of proposals in synthetic biology. One of our students just reached out to me and mentioned today that he's going to start his PhD in synthetic biology under my supervision, and he received some significant amount of funding from government, and then he's going to continue with his PhD as well. A lot more parts based on your application, right?
There's another set of problems about the project. I have sent it in a private chat. Have a look.
Mhm.
Can you see it? I'm an undergraduate biotechnology student at SLIT.
Are that if selected as a top student, would it be possible to join your company as an intern, considering our classes are held on weekdays? Okay, yeah, that we can definitely arrange a flexible work plan with our company once you say you are one of the top students performing in the project. So don't worry about that. Yeah, based on your availability, we can arrange flexible plans for you to be part of our projects.
Okay, so if there aren't any questions, we can wrap it up for today.
Yeah.
So you will get, let's wait 1 or 2 minutes more.
Okay, Sanjeev.
Okay, there's a question now.
So, what are the career paths available? Sir? What's the next step in the sense? Yeah, as I
I think you were cut off,
uh,
Lalinka. You have to say
the
that again.
finals, we wanted to focus on. Right. Okay. Am I audible now?
Yeah, yeah.
Sanjay, can you hear me well? Right. I'm well audible. Right. Okay. Now, the plan is to not to just stop from this particular once you are done with the course. We don't want you all to discontinue your work. If you have done some significant work, we will definitely guide you through how you would write a research article based on the outcome of your project. Because if you conduct simulation studies, now we can publish international research article, full length research outcome. So we can try submitting them in good journals and get it published. And also you can write review articles related to this area. And also we would guide you in how your idea is unique and patentable. We can guide you through the process of patenting your idea and claim your novelty of your project as well. So you can, we will always support you with those aspects throughout even after the course. Then will be exam held on. There's not going to be an exam, Fatima. What we are going to do is we are going to assess. We provide you an assignment. Assignment means you have to work on your own project proposal. We will we will assess your project's outcome, and then we will rank based on the novelty of the project that you have conducted, and we are going to select for the top 5 based on that. And there will be marking criteria for that. Um, you have to submit your project proposal, final project proposal document with us. course will end in last week of July. Examples, right? Okay. One of the examples is that let's say you are interested in healthcare applications. You want to come up with a diagnostic tool in order to, as I mentioned throughout my presentation as well, you want to diagnose some cancer condition. Let's say you want to diagnose some brain cancer, newly designed biological cell that can capture biomarkers from this particular brain tumor originated from the tumor site. And then these biomarkers present in your biopsy samples, your cell should be able to express a certain signal, detectable signals in order to sense for these biomarkers. These are diagnostic tools. And also if you have some interest in microbial related application, you can design new bacteria that can degrade microplastic. Also, you can design new bacteria that can clean pollutants in contaminated water bodies. Likewise, even there are interesting applications. You can design new, new. You can conceptualize new gene circuits that express in plants that plants can grow in, and microorganisms can be made modified in a way that they can grow. So many things. There are interesting applications like that, and also healthcare and environmental applications as well. Read those things in the applicational part of the topic. Yeah, Sanjay, we are going to share our PowerPoint materials with them, right?
Yes. So this is okay to share, no? This slide
Yeah, that's fine. Yeah, that's
okay.
fine.
Then we'll put it on the WhatsApp group. Or you guys can join the WhatsApp group.
Yeah. Right.
Okay. Cool. Okay. Then I think have we answered all the questions? Last one was by Sajida Manzoor. Have we answered that? Yeah, about the PowerPoint presentation.
Yeah.
Yes, everything will be accessible through our LMS, all the downloadable material and the recorded sessions, so you don't have to worry about that. All right, so we have about 7 days to start, 8 days actually, to start the class. So I hope that most of you here will actually jump in and take this opportunity as the first ever batch on synthetic biology by any institute in Sri Lanka. Okay, as a professional course, obviously, there are maybe some academic ones. This is the first professional course that is targeted at practical applications from this subject. So given that, it's time for us to say goodbye then. So thank you so much for joining. And if there are any questions, join the WhatsApp group. It has been SMS to you, email to you, as well as put on the chat here as well. So let's hope that you guys are joining this course and finding new pathways to brighten your future. Thanks, Dr. Lalinka, for the opportunity and the session.
Yeah, thank you very much, Sanjay. Have a very good night. Thanks everyone for joining this session.